Hi, my name is Ashley Bryant, and I'm a scholar of the SI Bridges program. My summer research project is genotyping genetically modified mice. The genes we're genotyping are forkhead transcription factors. There's approximately 50 forkhead genes in humans, and the two we're closely observing are FOXO1 and FOXO3. As you can see, they're closely related. They also aid in the role of somatotroph differentiation, which basically tells a cell to be a cell. They also both show expression in the pituitary gland. I've inserted an image here of my immunohistochemistry, and I won't dive into too deep about what IHC is for time concerns, but basically with fluorescence, we're able to highlight the expression of FOXO1 in this pituitary gland here. We use genetically modified mice for the role of observing these forkhead transcription factors in pituitary gland development and function. We also use a cre-lox mediated technology to create tissue-specific deletion or tissue-specific expression. And so what we're seeing here in this image is a FOXO1 wild type allele. We need to flox out this second exon, and we do so by inserting loxP sites and the introduction of cre, which then deletes that exon, and exon 1 and 3 are then ligated together, creating our new FOXO1 recombed allele, FOXO1 flox. We do this to promote pituitary specific expression because FOXO1 is also expressed in not only the pituitary gland, but also the heart and placenta as well. And if deletion were to occur everywhere, then the mice would die. In this experiment, we mated 2 mice together and they produced 10 offspring. The mother's genotype is FOXO1 flox, FOXO3 flox, FOXG1 plus. The father is FOXO1 plus flox, FOXO3 flox, FOXG1 plus cre. We could see that they're both homozygous for FOXO3 flox, so right off the bat, we know their offspring are also going to be homozygous for this gene. But we're going to see some differentiation here with the FOXO1, so that's what we're wanting to genotype. In order to genotype, we need to collect a biopsy sample, and so since they were embryos and they were so small, we took a snip of their tail, but you could also collect an ear sample. With our sample, we needed to isolate the DNA, create a master mix with primers and add it to each of the samples, run it through PCR for about 2 hours, and then run it through gel electrophoresis where we could form the migration of bands that can be visualized. Excuse me. In this lab, we don't necessarily use a molecular weight ladder. We have to utilize our controls, and our controls are previously genotyped mice with the presence of cre and without cre. I've created an animation here of what we're going to be looking for. Our wild type band should be this bottom band here, sitting at about 115 base pairs. That's going to be the plus flox. Our flox band is going to be sitting at about 149 base pairs. That's going to be our middle band here. And our null band, which is the presence of cre, no FOXO1, is going to be our top band at 190 base pairs. My last 2 wells are my positive and negative control, so they should have that. My positive should have the presence of cre, and you can see that here at the top band. My negative should not, but you can see it's there, so that's an indication that a potential error had occurred of contamination. My first 3 bands, you can see that the mice are FOXO1 plus flox with cre. And then these 2 don't have that bottom wild type band, so they are flox with cre. This is just a middle band, no top or bottom, so that's just flox without cre. And then my last 4 are flox with cre. In conclusion, my results coincide with my hypothesis, showing significant differentiation. 60% of the mice were FOXO1 flox, FOXO3 flox, FOXG1 with cre, and 30% were FOXO1 plus flox, FOXO3 flox, FOXOG1 with cre. And then only 10% were FOXO1 flox, FOXO3 flox, FOXG1 plus. To end my presentation, I want to acknowledge Dr. Buffy Ellsworth for being my mentor and amazing teacher and allowing me in her lab. Patricia Dawes, the grad student who has guided me and also been an amazing teacher. Dr. Laxmi Sagwan Barkdal for organizing the SI Bridges program and being extremely compassionate and helpful at any time. The SI Bridges for providing this opportunity, and NIH for funding this opportunity. And I'd be happy to answer any questions at this time. Thank you.